Okay. Hello, everyone. So welcome to all of our attendees for our next session on the intro to FlowJo, held by the Flow Cytometry Core Facility at Memorial Sloan Kettering Cancer Center. You might have just heard the phone ring. So as like most other people right now, I'm currently working from home. So pardon any miscellaneous noises that you might hear. And we hope everyone is staying safe right now, staying home, and everyone is happy and healthy. Okay, so with that, I should introduce myself. For anyone that is just joining us for the first time today, my name is Kathy Daniels, and I work as the Flow Cytometry Core Facility Manager at Memorial Sloan Kettering Cancer Center. You'll hear Rui Gardner also on the line. He's the head of the facility, and I'll say this throughout, but if anyone has any questions as we're going along, please feel free to use the chat function. Rui will be monitoring that, and as we go along, if anything pops up, seems confusing, you need a little bit of a clarification, please go ahead and just use that chat function. So again, today is going to be on cell cycle and proliferation. Okay, and some of the summary of the topics that we have today, we're going to be going over univariate cell cycle data. So just for example, DAPI, PI, and then we're going to be going over some bivariate cell cycle data with the incorporation of BrdU, okay? Then we'll review some of the different types of modeling for the analysis when you're looking at the univariate cell cycle, because there are different approaches to that. And then from there, we're going to move on and go to proliferation analysis and modeling. And we're going to talk about some of the, very briefly, show two different examples of different dyes that are available, because there are an array out there. Okay. Some important links that we want to stress to everyone that's coming and joining us. FlowJo has made a license available for researchers as they're working from home due to the current pandemic, and we thank them for that. And for more information, you go to flowjo.com and use this specific hyperlink with the COVID-19 license. So thank you to FlowJo. Then for the Flow Cytometry Core Facility website, it's fccf.mskcc.org. We post all of the video recordings of these sessions, in addition to other educational content, SOPs, news of our facility. And we also post these items in addition to announcements and other items on our Twitter, and our handle is FlowMSKCC. If you're interested in learning more about cytometry, we want to stress that you can also visit the ISAC website. It's our international society, and they have also made Cyto University free and available for people given the current scenario. So we thank ISAC for that initiative. And then Flow Repository is a nice source to get FCS files if you're working from home, but you might not have any files yourself to analyze, but still want to go ahead and take a look at FlowJo or any data analysis software. Some more important links, you'll see that there are two more that are here today that we usually don't have. And that's a link to the proliferation and cell cycle analysis platforms in FlowJo. So FlowJo has detailed explanations of some of the things that we'll be going over today. So I'd like to reference that for everyone to know that it's there to be able to get some more information. Then you have the Cytometry Part A journal that's available for research into cytometry articles. MetroFlow is our local New York, New Jersey flow cytometry users group. So I encourage everyone to go and check that out. We have 2 meetings every year. We have a vendor meeting and then our scientific meeting in the fall. So lastly, I just say thank you to David Gravano, and the link is at the very bottom here with the YouTube. What you'll notice at the very beginning, you had that text to FCS file with cell cycle and proliferation. And I thought that was pretty cool. So I've been using that a lot. And if you want to play around with some text or image to FCS files, you can absolutely do that. Okay. So the contacts to reach out to if you have any questions about this, there's Rui Gardner, who's the head of the facility again, at gardner@mskcc.org. And then there's myself, Kathy Daniels, and the email there is danielk1 mskcc.org. So this is a little bit clearer than previous slides where you can see that it's a 1 and not an L after. And lastly, we just wanted to thank our whole group. I can't stress it enough. They've been wonderful this whole time. They're on this webinar too, I believe. And I just want to say thank you to our group for being so great before all of this and now. Okay, so now we can go ahead and we can start looking at some data. Okay. So I'm gonna move myself a little bit down here, and I'm gonna go to my FlowJo icon right here on the desktop. And most people are aware of this, and I'm sorry if I repeat this every week, but I wanna make sure our new attendees are aware that FlowJo now mostly works from a portal ID login. Okay, so it's not a hardware-based login, but rather a portal ID if you're working from an individual license. It's a little bit different if you're working from a shared computer, but for individual licenses, you still have to go through the portal ID, okay? And as I'm signing in, for shared accounts, you also still need the portal ID, but that's a different topic. Okay. So I'm gonna go ahead and sign in, and it goes from unlicensed to a licensed version of FlowJo since I logged in with my credentials. Now, what we're gonna go over first is we're gonna go over some cell cycle data. So I have my folder here, and what you'll remember likely from last time, if you attended, is if when I open this folder up, I have a couple of other folders that all have cellular data in it from cytometry experiments, okay? And I've parsed this all out for what we're gonna be going through today. And the first thing that I wanna take a look at is some univariate cell cycle data. Now, this is the title of the folder. So what I can do is I can go ahead and double click and drag individual files in, or I can come over here and add samples, right? If I click that, it's gonna ask me where I wanna take them from. But my preferred method that I like to do is I like to just drag in that folder. And when I do that, I get the folder name as a group. Do you need that group? It's up to you. We've had multiple discussions about this, so I won't dive too deep into it. So I'm just gonna delete that because I don't necessarily need a separate group, and I like to keep things a little bit cleaner. So I just hit the delete key on my keyboard to be able to do that. And then I'm gonna X out of this file window right here. And then we're gonna start looking at our data, okay? So I wanna, before diving too deep, just say thank you to one of our researchers at Memorial Sloan Kettering for being kind enough to share some of this data with us. I won't call her out by name because I'm not sure if she wants that. She knows who she is, and we're very appreciative of her donating some of these files for analysis. Okay. So what I'm gonna do is I'm gonna go ahead and I'm gonna open up my first FCS file, and it defaults to forward scatter and side scatter. Okay. I can tell by eye that this was likely acquired on a BD instrument. But again, a reminder for everyone, because I think this is very important, regardless of if you've attended previous sessions. When I double click right here on this little circle, it's going to give me some keywords from those FCS files, or that FCS file specifically, okay? And when I come up and I scroll up, what I see here from the cytometer keyword, if I expand out, it's telling me that it's from the LSR Fortessa, okay? So just for people to be aware, that can be very important, and we might see that a little bit later on why that's going to be important. All right, so forward scatter, side scatter. What I first want to do is I want to gate on my cells of interest that I'm hovering over here with my mouse, and I want to get rid of all of this debris. There's not much debris or background in the sample, so it looks pretty beautiful. But what I'm going to do is I'm going to take this elliptical date and click and drag. And when I do that, it automatically comes up with the name of lymphocytes, and I'm just going to change that to cell scatter. I tend to veer away from calling or from naming a population of cells or annotating it strictly by scatter. I prefer to use CD markers, but for this case, this was just a cell line, so it doesn't really matter anyway. So I'm just going to call it cell scatter, and I'm going to take that and I'm going to drag it up to all samples. Okay. When I double click here, this is now going to show me from that same sample, which is specimen 1 at a 0 time point. It's going to just show me what's within that scatter gate. Okay, if I move this, it moves in real time with it. Okay, so any adjustments that I make are going to carry over there. And again, in here from the workspace, you'll see that that is no longer bolded. So if I say, okay, those changes I made, I didn't really want to make. If I hit delete there and yes on my keyboard, okay, all that happens, it keeps that gate. It doesn't completely delete it because it's owned by a group, and it switches it back to the position that it was automatically in, okay? Or originally in, rather. Okay. So again, I'm going to double click. Forward scatter area is going to be on the X, and forward scatter height is going to be on the Y. And what I'm going to do is I'm going to do my initial singlet gate. So I'm going to select the polygon option right here. That's my preferred method for singlet gating to start off with. But I want to stress that as we're doing this, just to go back to the basics a little bit, as you're doing these gates, feel free to explore the different options. So just because you're looking at forward scatter height area doesn't mean you can't look at side scatter height area or side scatter area width or forward scatter area width. Those parameters all work in order to look at and discriminate for doublets or aggregates of cells. The biggest or the strongest recommendation I would make is to explore all of those, because sometimes you'll see it pulls out a little bit better in certain parameters than others per cell type, right, or per experiment. So don't think that just because you have one favorite that it's going to apply to all. Just like dropping those little hints in for everyone as we go along. So then I'm going to try and see if we can do a double doublet, which we can, because the user was wonderful and went ahead and looked at a couple of different parameters here and enabled area and width for both forward and side scatter. So you see here, even though I did that initial singlet gating, there are still a few aggregates that are left over here. When we look at side scatter area width, you can also try side scatter height width. You know, they're kind of, and we can take a look at multiple of these in combination, okay? Don't worry, we're getting to the cell cycle very soon. It's just we want nice, clean data. So these are all things that'll help us get that nice, clean data. Okay. So then I can't have two gates of the same name. So I'm just going to do single cell side scatter. You could do two. You can call it whatever your heart desires. I'm going to hit OK. Okay. Drag that over. And then what I'm going to do is I'm going to highlight these two gates that I've created, and I'm going to drag it under cell scatter. Okay. Now what I'm ready to do is I'm ready to take a look at the cell cycle. This experiment, she was really just looking at cell cycle, so we didn't have to hone in on any populations in a heterogeneous tissue or anything like that. So some people might think, okay, let me just take a look at this. And then what I'll do is I'll look at my histogram of PI, because that's the dye that this user had utilized for her experiment. And if I do that and I come down and I select PI, and then here, you can simply select histogram. What some people might think is that you can come in and expand this out and use your interval gating to start setting a G1 and a G2 peak. And then from there, go ahead and define S based on that. That is something I want to stress that is not, stress is not best practice, right? Because if I do something like that and I come in and I make this gate right here, and I call that G1, and I call this G2. What winds up happening is we're visual creatures by eye, right? So we want to look at that and say, all right, this is roughly where it is, this is roughly where it is. But when we think about the histograms and univariate data and how that falls, if you follow my mouse, what would happen in a normal distribution is this is going to kind of come over a little bit, and this is going to kind of come over a little bit. So we don't have the ability to set those gates by eye to be able to accurately determine with a univariate set of data where my G1 and G2 and S phase actually lie. So I just wanted to bring that up and show you something that some people might be trying to do that probably wouldn't be the best practice when trying to analyze cell cycle data, because we're limited in our capabilities as humans to be able to pull out that univariate data. Okay, so what I'm going to do instead is I'm going to click on this population, and I want to look at the biology, right? But I skipped a little bit too fast, because actually what I should do very quickly is run through and just check those gates before I go ahead and start looking at the cell cycle modeling for all of this data, okay? So to do that, I think I told everyone this trick before, but if you hit Shift and hold down Shift on your keyboard, and if all of these plots are listing the same sample, so specimen 001, 0 seconds, dot 0 or underscore 006 FCS, once I start scrolling through, I'm looking cohesively at each sample for all 3 gating strategies, or all the gating strategy for all for the sample in one picture, and I'm able to just quickly verify that that looks pretty good. Okay, I'm not going to get too crazy with it. I'm sure I would have made some modifications if it wasn't just a training session, like maybe kind of hone in here and pull that in a little bit, but I'm not going to worry about those small changes today. Okay. All right, so then, like I mentioned, I can close this out now, and I'm going to go back and I'm going to start looking at the modeling for cell cycle. So this single cell side scatter right here, that's the population that I want to start looking at the single cell data, or start looking at the cell cycle data on. So you'll see there's this little biology tab right here, and if I click on that, there are different options. So there's 2 different options that we're going to be utilizing today. The data that we're looking at now is cell cycle, not proliferation modeling. So I'm going to go ahead and select that cell cycle right here. Okay. So what's wrong here? It's telling us right now that we cannot calculate the model, and it's unable to calculate the model based on the inputs. Why is that? Okay. If I was in a classroom, I'd ask for someone to pop their hand up, but I'm just going to tell you guys, the DAPI parameter was open, but from what you saw previously on the gating that I manually put in, her staining was actually with PI. So if I select PI, this looks a lot more like what we'd expect, okay? So this actually looks pretty good, I will say. I'm gonna walk you through some of the different things that are actually coming up here, how to analyze this data, and how to make any changes as you see fit if things might not pop up exactly the way you want them to be. So the first thing that I'm gonna do, I'm gonna kind of expand out on those 2 options of model and constraints, that we can talk about them a little bit deeper as we go through. Okay. The first thing I do is I take a look at this modeling and I say, right away, is the modeling fitting the data? Okay. Now, when we ask that question, it can be a little bit hard to determine, you know, just by eye. And what we want to do is, if we look at this, the root mean squared value, when we look at this, the lower that value is, the better the modeling is at fitting the model itself to the raw data. So if we have a very low value, which here it's 1.18, and you might say, okay, what does 1.18 mean to me? Does it need to be 0? Does it need to be 0.5? The guidance by FlowJo is actually that value, they take it as somewhat acceptable if it's under 10. So they're somewhat confident in the data if it's under 10. So this value of 1.18 is actually pretty good, okay? So this modeling has actually done a pretty good job at fitting this data to a cell cycle model, okay? Now, the Watson model, just to give you a very brief review, what it does is it actually assumes a normal distribution of the peaks of either G1 or G2. And so it's that normal Gaussian distribution. And then what it does is it assumes about a 1.75 increase, like the value of the G2 is about 1.75 of that G1, if I'm correct. And it adds up how many events would be in that G1 and then that G2. And then it subtracts that from the total number of events, and that's how you wind up with your S phase, okay? The Dean-Jett model is a little bit different. So if I click on that Dean-Jett modeling, what you'll see once I click on that, it automatically is going to switch to that modeling technique. And here on that RMS that I was mentioning before, the RMSD, that number has gone up. Okay, so that means that fitting is not as good as the Watson model. Now, you feel free to play with this and feel free to go between models and see which one fits your data the best. I'm not going to make a recommendation here on which one you should use. The Dean-Jett actually fits through a mathematical function, which is a polynomial, to be able to get that modeling of the data. Can I go into the deep mathematics of that? Unfortunately, I cannot, but we can always have an offline discussion about that if needed, okay? I just want you guys to understand that there are different methods for modeling this data. Okay, so for the purposes of today, I'm gonna go back to the Watson because I think that, and not only think, but I see that it's fitting this data a little bit better. And what you can do, say it didn't look quite as nice as this. I'm going to show you some different options that you can utilize to try and optimize that that RMS. Okay.
Kathy, before you do,
Yep.
let me just remind everyone that if they want to ask any questions, to feel free to write them down in the chat, and I'll be more than happy to ask them to you.
Awesome. Yes, that is a very, very good point. You know, and we'll be happy to answer any questions and do our best as you come through with them. Okay. So, like I was mentioning, what we can do is we can actually make some adjustments as we're going along to be able to try and fit these peaks a little bit better. Okay. So one of the things that you'll see in this plot right here is that I have the option to put in an interval gate to set a range. So it's telling me it's a minimum and maximum range to define inclusion or exclusion of a single dimension. But for our purposes, if I just click on that, what I can do is I can come in here and I can say that, oops, sorry, do it again. I can constrain that to be my G1 range, okay? Now when I do that, what you'll see is this G1 peak here now has been set at a range, and it's not unconstrained and strictly just based on the modeling, okay? If I go back to range, what happens? Everything has disappeared. I have no modeling, the data's gone. Please don't worry about that. You can simply just go back to unconstrained, or if you hit range, what you could do is just define that range, okay? And say this is my G1 range. You might have populations sometimes that pop up that are a little bit messier, that you have a lot of background, a lot of crud that might come up. It's a scientific term, crud. And that was a joke for anybody. And when you're doing that, it might have a hard time finding it. So you might need to give it a little bit of guidance, okay? Now, the other thing that you have the option to do, you'll see that the RMS is actually pretty good still when I set it at that range compared to the unconstrained. So there's really not much of a difference there, okay? But the other thing that you have the option of doing is setting the CV for the G1, okay? For this data, I'm just showing you how to make those changes, but for this, I actually wouldn't recommend doing it because it looks very nice, okay? For the CV, if I come in, and what I can do is I can either say it's a value of n and define that CV myself, or I can come in and say that that's my, it's going to be the same as my G2 CV. What is my G2 CV and how do I know? Okay. Up here, it's giving me a lot of statistics. So based on everything that it has modeled right now, it's telling me my G1 CV is 5.75 and my G2 CV is 5.63. Typically, because we're looking at this data in a linear fashion, because it's DNA and it's just G1 or G2 and it's double the amount, or you have polyploid that are continuing to double, right? The CVs of the data are somewhat similar between the G1 and the G2 peak, okay? So if I come in there and I say, all right, you know what, I don't want my G1 CV to be 5.75, I think it's a little bit higher than that. If I hit N, it automatically defaults to 1, and what happens? This does not look great anymore. Even though that RMS is coming at a very low level, we have no G1, and we know that that's actually not correct. So if I start adjusting this value, the CV of 2, or the coefficient of variation of that peak, right, and how tight it is. Ooh, I don't want 33, that's not gonna be good. 3. I want you to see that the G1 is coming back. We're seeing that population, and this RMS is going down, okay? 4, it's going down even more. I opened up an app I did not want to open up, so I'll be Xing out of that in a second. And you can see that's an app that's not utilized on this computer. Okay, and as I go up and up, what are you seeing? You're seeing that that RMS is actually going down, okay? So this is ways that you can go ahead, and if there's a sample that's not fitting exactly right, you can go ahead and make those minor modifications when the modeling itself is having a little bit of a problem, okay? Over here in the graph option, you have draw model sum. If I uncheck that, it's not showing us that line that's showing the, that pink or fuchsia line that's showing us the fitting of the modeling. The draw components takes away the G1, the S, and the G2. And then if the components are there, but you don't want them filled and you just want the outline, you can do that as well, okay? So just to let you know that those are all there. The one thing that would be nice if it was here would be the ratio of the mean of G2 and G1, okay? Because what we expect is we expect to see roughly double the amount of G2 as far as the mean as we do for G1. I'm going to show you how to do that with stats very quickly, but that's the only thing that would be nice if it's in there, but we could very easily add it into the stats or stats table. Okay. These options are also here for G2, right? So you can say that you want the G2 to either be a range that you set. I don't know why that's happening. Okay, might have been because a question came through. So it could either be a range that you set through that interval gate, or it could be the G1 times N. If I wanted to set that up and make adjustments there, right now, if I say it's exactly 2 and exactly double the mean of G2 that it is for G1, you'll see that does not look great. So it's likely a little bit lower than that. And if I type in 1.95, you see it gets a little bit better. Oops, click the wrong thing here. If I type in 1.9, even better, right? So these are all things that you can do to kind of adjust and make your fitting a little bit better if you need, okay? If the data is nice and clean and it looks pretty good, you shouldn't have that much of an issue, but just so that you know, these are all options available to you. I'm gonna go ahead and set this back to unconstrained. And I'm
Kathy, while you're doing that, one of the questions that just came up is regarding applying the same settings to all samples, or can we adjust them for different samples?
Great question. So what would happen is, as I'm going along, the next thing that I was actually going to do, so that was a perfect timing for that question, is come in here and you can right click and you can copy that analysis to the group, right? Or you can drag it up to that same population. Now, when you do that, if I had made those modifications, right? So say I made the G1 peak or the G1 CV 1, and then I drag that up to all. I believe that should carry over, and you see right here, it does. So as I go through all these samples, they're all gonna be incorrect, but I'm doing that to show you the drastic, because it's so drastic and how you'll see that change, how that adjustment will carry over to all, okay? Now, say I'm like, you know what, that definitely does not look good. The CV I'm gonna let be unconstrained, and I'm gonna let the Watson or the pragmatic modeling function go ahead and take care of this. What I can then do, and you'll recognize that this cell cycle little tab right here is no longer bold. That's because I made a change to it, right? And all the rest of them are going to be back at 1 for the CV of that G1 peak. So what I can do is I can right click and I can copy the analysis to the group, or I can again drag it up, okay? So I'm gonna show you the copy analysis to the group because I don't think I've actually shown that yet. And I'm gonna replace the whole group. And then as I go along, it actually went ahead and made those adjustments. So the second part of that question was, can I go ahead and make changes per sample? What I would say, you might run into a circumstance where there's a certain treatment where you have a lot of background, you have a lot of maybe sub G1 or apoptotic cells that are popping up, and it gets a little bit messy and it's hard for the modeling to find. In that scenario, you might be able to go in and make an adjustment and put in a range of the G1. But I would be careful. What I'd always suggest, and you'll see we always make all these little comments, what I would suggest to do is optimize your experiment as much as possible, right, from a technical standpoint, so that the modeling should hopefully work for all of your samples. If you do have to make modifications, understand, you know, there might be some implications to that. And Rui, please correct me if I'm wrong or anything like that, or if you have any, not if I'm wrong, but if you have any added comments for that. But what I'd suggest is if there's anything that's way different, try thinking if there's a biological reason for that and it makes sense that you'd have to make some of those adjustments. If not, there might be a technical reason why something had happened there where you'd have to make some changes and adjustments. Sometimes I've seen people come and it might be their first time doing Cell Cycle, and this peak that's right here at about 50,000 might be shifting up and down and up and down, and they feel like they have to adjust each sample. That is likely because they didn't keep the correct cell number or the same cell number per tube. And then and if it's not at saturation with the concentration of the dye, what's going to happen is you're going to see shifts of that signal. So as much as you can control your experiment from a technical perspective.
Yes, yes. Yeah, it's important to note that, you know, we want to be as consistent as possible, both in sample preparation, in acquisition, and then in analysis, right? So we don't want to change too many things from sample to sample without a strong reason, right? Without a good criteria for that. What Kathy is saying here is that you can have some changes, some biological changes that, you know, one model won't be able to fit all of the samples because of those biological changes. The thing is, we don't know, right? And so again, we want to be as consistent as possible and avoid that. But if you see that the fitting is much better for one sample when you change the model compared to all the other samples where you didn't have to change the constraints, then really what's important is to have a good fit, right? And if you have a good fit, then the, you know, you're getting the best data out of the modeling. So, you know, I would say I agree with Kathy. We should try to be as consistent as possible, but there may be some reasons that that will lead us to change a little bit the constraints from sample to sample.
Yeah, especially if they're, you know, with treatments and stuff like that, you'll see some changes like I mentioned. And I do have a data set that I wasn't going to bring up during the main session, but I'm pretty sure that I have a sample where it does adjust, and there's like a pretty big increase in the sub G1 peak that you'll see. And when I say sub G1, right, this G1 peak right here in a nice controlled sample, everything below that would be considered sub G1, and sometimes you see a hump here, and what that typically is referred to as sub G1, or cells that are maybe starting to have DNA fragmentation, they're starting to undergo apoptosis, you might see them there, and that might throw it a little bit. So if there's time at the end, I could always bring that in after we do our second analysis and proliferation.
Right. Just let me just ask you a question, because a few people here have brought this up. You know, you went immediately from a simple data, and some people here asking, you know, should we address quality control of sample, like looking at the time gate? Do we need FMO controls and compensation? And even do we need to gate out the dead cells? You know, the typical things we do with immunophenotyping.
So my suggestion, I have a couple of suggestions there. So for time gate, we brought it up briefly during another session, but that's something that's always a good practice to do, regardless, to take a look at a parameter versus time, right? So I can show you how it looks like when if we wanted to check those out. And there are certain certain different plugins that you can use to help out with that in FlowJo, and that might be one of our future sessions, utilizing FlowClean or something like that, that's available to help us out. That's something that you always do want to be mindful of, so you can do a quick check of that, right? So if I open up a plot and I look at time on the x-axis, you can see here everything's pretty stable, and I can do a quick run through and go through all my samples and be on the lookout for any drops in signal, whether it's fluorescence or scatter or anything like that, that might indicate that there's either a change in laser delay, right, or a change in laser intensity if it was based off of PI, right? So you see here, there might have been a little bit of a blip. They might have stopped the sample. They might have paused it for a little bit. They might have needed to re-vortex. This I wouldn't be super worried about, but if I was, we can always set a gate and exclude that. But checking the quality of the sample by time is always a good suggestion. Okay, now talking about viability dyes, okay? Now, if you're worried about, I've actually made this suggestion, I think this user had actually done that you, if they're fixing and utilizing, if they're fixing, they can always go ahead and use fixable live dead dye and stain their cells with fixable live dead dye, then do the fixation and then do the cell cycle analysis. This way they can pre-gate out their dead cells, okay? That is an option. It's not something that people always utilize, but the more you can increase the quality of your data by ensuring what you're looking at are the live cells, we would suggest that. There's also some live cell cycle options that you can utilize that you wouldn't necessarily need that. So that's my viability suggestion. And then as far as FMO controls go, it depends on what you're looking at. So for this data set, I don't need to look at an FMO, because when we think about FMOs, that's the contribution of multiple fluorochromes, and you're having an issue with some spreading of the data, and you need to be able to define where your positive and the negative populations are. For cell cycle, if you're just looking at PI, right, or you're just gating out the dead cells, excuse me, and then looking at PI or DAPI or whatever it is, you do not need to worry about an FMO, okay? That's gonna be if you're doing a panel of maybe a couple of colors, there's some spread, and then you're needing to better define positive and negative. Cell cycle is a little bit different, and you're looking at the linear data, typically in 1 or 2 parameters. Okay. Any more questions, or we're good on that front? Okay.
We're good, we're good.
Perfect. Okay, so I'm gonna close out that, and those are great questions. Keep them coming, guys. So now I have that cell cycle data for everything, so for all my samples rather. So what are some cool things that I can do with that? One, I'm actually gonna go ahead and open this up again, because I actually wanna show you something else. What we can do that I forgot to show you, in the modeling, you can copy the gates in the workspace, okay? So you see, once I selected that here, you see the G1, the G2, and the S phase. So even though my eye was close, right, it's still off by a couple of percentage points, and I wasn't able to super accurately define the G1 and the G2 peak, which is why I wanted to show you that here, okay? So I can go ahead and delete those old gates that I made. But it does give me the option to copy those gates. And then if I opened the layout editor, what could I do if I wanted? I can go ahead and drag in the G1, the G2, and the S phase as overlays. Or what I can do, I can just drag in the cell cycle analysis. And when I do that, I can expand this out. What's nice about this, and the reason I might like that a little bit more, it's showing me the cell cycle, it's showing me the fitting and everything like that. It's showing me the different components with the fill. And it's also giving me my statistics all right here. It's telling me what percentage is less than G1, my sub G1 population, what might be considered polyploid if you're looking for that, which is greater than P2, the CVs of the data, and it's also giving me the means, okay? Very quickly, we already went over batch reports and everything like that. We need to show the bivariate data and then the proliferation data. So I'm not going to focus on this too much. But what we should expect, right, and what I mentioned before is that ideally what we should see is that the G2 should be double the amount of G1. So if I open this table editor right here, what can I do? I can say for this G2, I want to go to the workspace and I want to look at statistics. So if I look at the mean of PI from G2, and I look at the mean of G1 for PI, okay, and I could have left that statistic box open, I just wasn't thinking. What I could then do is I can hit Command on my Mac, or I believe it's Control on the PC, and I can drag those into the table editor. And then I can go ahead and I can edit. I can add a column. You guys know I love columns if you've been with me a couple of sessions now. And you'll also see that I can cut my own hair at this point because I've done that in the past couple of weeks from home. Poor attempt at a joke, but I did. Okay, so apologize, G2. Okay, so we have G2, and then what I'm gonna do is divide that by G1. Very simple. All I want, and if I can title this if I want, G2 over G1, call it whatever you'd like. If I come back to the table editor, I can create that table. And because I only put in those populations for that first sample, it's only going to show up for that one. But you can see it's pretty close. It's 1.85. So we have roughly double the amount of PI signal and DNA in G2 than you have in G1. So that's just a nice little QC method for yourself to be able to make sure everything worked okay. I'm going to close that out. I'm going to save this workspace, and by going Save As, or we're just going to save rather. And this is going to go into the session from today, the Univariate data. So always save your workspaces where your FCS files are. And the other recommendation I'll have for you quickly is make sure that you go ahead, if you have files on the server, try and transfer them to a local drive before you start analyzing, or else you'll see that little circle going and going as it's trying to read from the server and back to your desktop. So that's why I always work from the desktop whenever possible. Okay, so next we're going to go over some bivariate cell cycle data. So I'm going to open up this folder again. I'm going to drag in bivariate cell cycle, some bivariate cell cycle data. Okay. Now, when I bring this in, I'm not going to analyze all of these samples, okay? I'm just going to analyze one or two because I want to show you an important point here. Okay. If I open this up, I'm going to do some of that gating we discussed before. So scatter, and I'm going to go a little bit quicker. Okay. I mentioned this in a previous section. There's an option in the preferences where I can adjust where this annotation goes so I don't have to move it each time. If anyone wants to see that later, just let me know, and we can do that after. So as I'm doing this, I'm going to just let you guys know, there's a reason when I talk about bivariate, right, cell cycle data, I'll spoil the surprise. It's going to be, that's when we try and incorporate another marker in there for us to be able to pull out the cells that are in different phases of cell cycle, right? So for this data, we actually have BrdU, and the Alexa 647 is an anti-BrdU. So the BrdU is a nucleoside that we'd put in into the cells and let them incubate, and then after we can go ahead and after we do the viability and we do surface staining, whatever's necessary, we can come in with an anti-BrdU antibody tagged to a fluorochrome to be able to pull out cells in S phase. So that's what we're going to see here. Okay, I'm going to go back up to this is the control right here. So if I double click on this control and I look at PI, that is a negative control. Okay. So if I look at PI and a histogram, that does not look so great, okay? So you can see for all of these, it's not a super clear signal, right? So this one might be the best one. And the recommendation that I would always have is to make sure you have a nice control population, but sometimes your staining might not look so great. So how can the second marker help you out? Close all of that. I'm actually going to delete this control. Actually, I'm not. Take that back. Okay. What I'm going to do is I'm going to take a look at the cell cycle there. Okay. I'm going to look at the PI. It's not looking that wonderful. Now, by not wonderful, it's not showing me any modeling, right? I can come in, I can put in a constraint, and I can set that as G1, right? And I can try doing my best by saying G2 is going to be G1 by maybe 1.7. I can start making some assumptions here and try pulling out some data. 1.6. Can I get this where it looks okay? Well, I'm having a hard time with this, right? I might have to spend a good amount of time pulling out where's my G1, where's my G2. This data is not looking the best. Or what I can do, because I'm not going to spend too much time on this, I can open that sample up and I can look at PI. And then I told you my Alexa 647 was against BrdU. Now, what do I see here? I see something that looks much, much better, okay? The first thing I'm going to do is I'm going to use this transform button and customize the axis, okay? And I'm going to go ahead and try and make that scaling on the negative end a little bit better. Sometimes it just scales up and that's okay. The population's a little higher up and that's okay. And I'm going to make this a little bit bigger so that we can all see a little bit better, okay? Now, when I do that, what do we see? We see a very clearly defined Alexa 647 high, and then we also see this population here and this population here. So what is that, right? The BrdU is incorporated by cells as they're undergoing the division, and they're in the S phase, right? So those BrdU Alexa 647 positives are our S phase. So by doing that, what we're able to do, I could do a polygon gate. I probably should have just done a rectangular gate. But if I select that gate, I can say these are my S phase. Not phage. We're not looking at virus right now. Phase. Okay. And then what I could do is I could take a look and draw a gate around my G1 and my G2. Okay. So by doing that, I'm able to get bivariate data, I'm able to get cleaner data, and I'm able to get a really good reading on, and more accurate reading on my S phase, and get more accurate statistics by looking at another parameter. So that's something that I would suggest, and that this researcher at Sloan had started doing that pulled it out much better, okay? I can then go ahead and apply that up to the rest of them, and start looking at this and making minor modifications as I have to. But if I try and compare this right here to a histogram, it's going to be way harder for us to pull out that S phase as things are starting to merge a little bit closer together just with the PI than it is with us looking at the addition of BrdU. I do have another data set with this where I can compare, we can get the PI or the DAPI rather, cell cycle modeling to work, and then we can compare numbers from that to the actual incorporation of BrdU, and we can see the differences in that and those statistics. But what I'm going to do is I'm going to actually ask Rui's advice on that one. So do you think we should see that data set, or should we move ahead to proliferation given the time?
I think unless people ask for that, I think we should probably move on.
Right? Okay.
Can I just ask you a couple of questions? So one of them is that so somebody was asking about the doing the double discrimination with the fluorescence, because that's something very typical to do when analyzing DNA cell cycle.
Yes, that actually is something you can absolutely utilize if you have the height an area or with selected, typically on Viva instruments, you just have to be careful about how you select it. But if you have those parameters selected, please feel free, and you absolutely can take a look at height versus area. So if it's PI height versus area, you can do that. In that data set that I had, I don't believe that I could. So the singlet determination by the height and the area is what we can do for that data set. But we often sort nuclei for our users, and we take a look at the DAPI height versus area. And another little tip to help everyone out, when you're taking a look at your cell cycle data, what we do in our facility before we do any of those sorts is we throw beads on to make sure we can check the CV of the QC beads on our instrument to quality control it to make sure it's nice and tight, because reliable data not only relies on the technical aspect of your experiment and biological, it also relies on the instrument performing up to its best standards and making sure it's nice and clean, that the PMT is nice and the detector is nice and linear, and also it relies on and the alignment of the laser and all of that to make sure everything's right where it should be. So keep that in mind, and if you're a user of a core facility or an SRL, you can always ask those questions, whether it's here at MSKCC, not my house, I have a cytometer here, or whatever facility you're in. Feel free to ask the people that are running your facility, or you can always reach out to me myself if you have your own instrument.
Yeah, and very quickly, because somebody was really excited to know, I think you showed this in another session, but where do you change the annotation location in the preferences?
Oh, yes, okay. I'll show you that with the proliferation data, because that's something I also enjoy very well. So I'll bring that in, and I will be sure to show you that.
Okay, thank you.
So before we do the proliferation and actually bring the samples in, I'm just going to briefly show you 2 or 3 slides just to show you what some common tips for proliferation, and I'm also going to show you some of the stats that we're going to be seeing and what they mean. And remember, we're going to be distributing these slides to everyone who registered to this class. Okay. So I'm not going to go into slideshow here, but I'm just going to briefly show you. This is a flow post-it that was put together by Barbara Olivera in our group on cell proliferation, if I can speak, by flow cytometry. So it has some nice tips on how to actually go ahead and look at cell proliferation by flow. And it talks about the different dyes available. You have the nucleoside analog incorporation with BrdU or EDU. That's something that we just looked at with the cell cycle. Then there's also dye dilution proliferation assays, such as CFSE or cell trace. We're going to be looking at some of those today, where they covalently bind to intracellular molecules like amines, right? And then they're inside of the cells and they can't get out, right? So they remain in the cells, and as they divide, each daughter cell has about half, okay? So that's very important to remember. If the parent cell has 100 molecules, you would expect each daughter to each have half, roughly. Okay, and it should add up to a total value of like 100% or 1. Okay, the other type is cell cycle associated proteins such as Ki67 or PCNA. If you're interested in any of these flow post-its, you can visit our website or the Twitter that I mentioned earlier. Okay. We have some statistical definitions. This is all taken directly from the FlowJo website, and it's referenced down here. It's from their version 9 website. It defines everything really, really nicely. So I took it directly from the site. I can't take any credit for this. We're going to be focusing mostly on the division index and the proliferation index. And we're also going to be looking at some other things like peak ratio, okay? So I'll talk about it very briefly, but the definitions are here. Okay. And I do have an example we can go back to later that discusses how we kind of get to some of those numbers if we have the time. I'd like to get to some of the data, but it just goes through some examples of how we would get the division index, the proliferation index, and also how we understand starting cell numbers. So for people that want to stick around later, I'll go ahead and go through some of those statistics and the basics, but just so that you know, we're going to be going over some of that at the end if you stick around. Okay, so open up a new workspace, and then we're going to show you our proliferation data. Okay. So if I open up that folder, I want to actually thank John Quinn from FlowJo, who helped me out and sent some proliferation data, and also Stella Chun from Thermo, who also sent me an FCS file showing their cell trace violet. So I'm going to show you both of those today. We're going to start off with the proliferation data from FlowJo. So when I drag that in, what I'm going to do first is I'm going to show that user who, or the researcher who asked the option to go ahead and change annotation. So if I go into preferences, right, what I want to do first is I want to go into gates, okay? When I go into gates, the annotation position, right now it's showing as inside. So when I draw a gate, what's going to happen is it's going to, say the gate's right here, it's going to put it right in the middle, okay? If I set it above, it's going to set it towards the top of that plot. So I'm setting it as above, and I'm going to go ahead and hit OK. Now, once you change those settings, moving forward, it should work out for you. Sometimes when you make changes in the preferences, you have to quit out of the program and open it back up for it to save. I'm not sure that that's the case with annotation off the top of my head. So if it doesn't show up exactly like that, that's why. Okay. So we have our unstemmed sample right here. And if I come in, I'm going to draw a proliferation gate, okay? I'm not exactly sure which cells of my interest. I think that's some debris and crud. I think those are likely my cells, if I have to make the guess. But this sample actually is stained for multiple markers, so I'm going to be able to pull out the cells that I'm looking for. I'm going to call that my scatter gate. Okay. And I'm going to bring that up to proliferation data. Okay. We've discussed in previous sessions, you don't have to start off with a scatter gate. You can always start by looking at your viability versus CD marker of interest. That is something I like doing. That might be great for this, but I'm just going to leave it as is because I just already did it. Okay. And you can see that annotation is now above. Okay. If I scroll over, what can I see? I can see there's a change, and there was a stimulation which can oftentimes change the scatter profile of a population, okay? So please be mindful that it is okay to adjust that scatter gating as you go between, you go between unstimulated and stemmed, because as cells become activated, as they're starting to proliferate, there very well might be a significant change in the scatter profile. And the other thing here that you'll notice is the side scatters in log. That is something that researchers commonly do. There's nothing wrong with it. If I changed it to linear through this transform button, I am not going to be able to reliably gate there. So I'm going to leave it in log. Okay, open that up. Now we can take a look at the singlets. So forward scatter width, forward scatter area, single cells, drag that up. Both of them. Okay, I'm just gonna, again, for sake of time, just do one singlet gate. I think we should be okay, understanding that you can do two. Okay. Now, next thing I'm gonna look at is I'm gonna look at the 7AAD, which is the viability here, and then I'm gonna look at CD4, all right? Now, we always want to reference back to an unstained or an FMO. Because we're working from home, I've been very reliant on the generosity of people to be lending me data to be able to work with you guys on this. So I unfortunately don't have all the controls that we might always be looking for. But what I can say is I can say, all right, I see this population here. Those would be, actually, yep, my CD4 negative. I also see my 7AAD positive, negative, and then I have the double positive. So what I want to gate on is my live CD4. I'm going to call that live CD4, okay? Because for this assay, what they're looking at is they're looking at proliferating T cells, which is a common application. Okay, let me go to the next one. I can modify that gate. And for this experiment and for this sample, I'm just showing you an unstemmed versus a stemmed. Okay, close that up. We're happy with all of that. And then what I'm going to do is I'm going to go ahead and I'm going to take a look at the proliferation. So again, I'm not going to go too crazy into this, but like I said earlier, if I look at the proliferation dye, this one's E-Fluor 670. If I look at that, I'm just in a histogram, what do I see? I see that unstimmed population is nice and high. It's up there, and you always want that control to be able to define where your undivided cells are, okay? And then when I go to the next sample, which is one of the stemmed populations, you see a very nice pull out of different populations as they're dividing, okay? I cannot draw gates on that to be able to pull out how many divisions there are, or anything like that. That is something I'm not even going to try and draw gates on. Some people might look at data via, you know, versus another parameter or anything like that. I still cannot pull that out. So we want to utilize, again, that biology tool or biology option right here. And then instead of looking at cell cycle, this time we're going to look at proliferation modeling. So I'm going to start off with my unstemmed. Okay. I'm going to select that. And I want you to be very careful, because it's not every single time that it recognizes which parameter that you're supposed to look at, okay? So what I want to do is I want to change this GFP to the eFluor 670 dye, which I know is my proliferation marker. Okay. Once I do that, I know this is my unstem. Why does that matter? It matters, again, because that's where I know I have my population of cells that are undivided, because I haven't treated them with anything to stimulate them to proliferate. So I click this here, right there. I drag it along, and I'm fixing that as peak 0. I'm telling the software, these are my undivided cells. Then what I can do, I know it's been an hour because the clock is going off. I can go ahead and I can copy that analysis to the group. So this is pretty straightforward. If I come to the next one, you'll start seeing that it starts to model that data, okay? Does it look great right now? It does not. I'm gonna walk you through some of the things that we wanna look at and how to adjust this so that it's actually fitting the data correctly. The first thing that I actually wanna go ahead and do is I wanna take a look at how many peaks am I able to see by eye. So with the mouse, I can see 1, which is my cells that still have not divided, 2, 3, 4, 5. So what we want to do is we want to take that 5 and we want to add 1. And then what we can do is we can tell the modeling, let's look for 6 peaks. Okay? If I hit 6 and then hit enter, look what happens, okay? That data looks much better. We can still go ahead and make some modifications, but that data is now modeling really, really well with this data set with these proliferating T cells. Now, if I go back to 4, I should have pointed out to you, we also have this root mean squared value for the proliferation modeling. Again, for people that have been with us since the beginning, that root mean squared, the lower it is, the better it is at modeling and fitting, rather, the raw data to the model. Okay, so the lower that is, the better your modeling is at being able to fit to the raw data. Okay, so keep an eye on that. That's 3.76. I go to the 6, which is the 5 peaks that we can visually pull out, plus 1. Okay, now this looks pretty good to me. I'm going to show you some tricks that you can use to go ahead and modify this and kind of adjust it so that it's fitting a little bit better. And before I do that, I'm going to show you some options in the graph. So here in Draw Model Sum, it's very similar to Cell Cycle, where if I put that in, it's showing me how that modeling sum is based on that red line as it's going through. Okay. You can also show the peak numbers. And it's going to tell you this is my undivided, divided population, or generation 1, 2, 3, 4, 5. Okay. I'm going to take that away for now. This can be a little bit distracting. And one of the things that, that you can do first is take a look after you've adjusted the number of peaks at that peak ratio. Okay. So as I mentioned before, what do we expect? We have a starting population that's undivided that has a certain amount of dye. Okay. For example, it has 100 molecules. As those 2 cells divide, it's physically impossible, or physically impossible for those 2 populations to each have more than 50%. The peak ratio, if it's above 0.5, is saying, all right, my two daughter cells each have 51% of that starting amount of dye. That's impossible. So we know that that fixed ratio, or the peak ratio, cannot be anything above 0.5. So I'm going to select here to fix the ratio, and I'm going to set that to 0.5. So I always want you to be on the lookout for that. Now, when I do that, what do we see? The root mean squared went up a little bit, but this population right here has shifted, and we're able to have a little bit of a better starting undivided population. So know that I want you to take a look at that and make sure that your fixed ratio is correct at the start. If I change that to 0.4, it's going to look horrible. So very minor changes have a pretty big impact. So I want you to be aware of that. I'm going to change that back to 0.5, and I'm going to look at the values that best fit this data. The next thing that I want you to be aware of is that it is important whenever possible that you have a population of cells that's stained for everything else with the exception of your proliferation dye. What's the reasoning for that, right? So someone before mentioned FMOs. So we have a couple of different fluorochromes in here, and you might see a little bit of spreading, right, for us to define where our negative and our positive are. And if I had a sample that had everything in it with the exception of this eFluor 670, what I would be able to do is I'd be able to look at that population and gate on the negative there and look at the statistics and get an idea of the background. Because having an idea of the background is going to help us to define where where it would be possible to have our eFluor 670, okay? And pull out between eFluor 670 positive and negative. So I don't have that for today, but what I want you to do is I want you to use this example as an example for why you would want that, okay? So if I choose fixed background, and I say, if I had to make a very educated guess, not best practice, I want to stress this, and I'm using this example so that you know what we'd want to do is to include a sample that did not have the eFluor 670. What I could do is I could say, all right, I think this MFI is somewhere around 500 that's where our negatives would be. As I do that, what do you see? You see this modeling fitting a little bit better because it's having a better understanding of where the background would be for that sample, okay? As I make that adjustment and I go up to about 650 or so, what are we seeing? We're seeing the data is fitting much better, and we're seeing that root mean squared value go down about 1.22. Okay, so you can adjust this data. You can also go around and adjust the peak CVs. I think that these CVs are fitting actually pretty well. It's telling you right now that the CV of each peak, or the coefficient and the tightness of those populations, are around 3.33 each. If you wanted to adjust this, say I wanted to set it at 5, doesn't look great. They're too broad. It's not fitting the data set that we have. If you set it at 3, it's a little too narrow. That might not be loving it too. You see how it starts to kind of go way too small there. So we can always play around with it. We can fix the CV, we can unfix the CV, and you see once I unfix it, and it sets it at about 3.33, that root mean squared, where the fitting of the raw data and the modeling is working much better now, okay? So these are all adjustments that you can make, and you can make them to go ahead and ensure that the data is fitting correctly, okay? So there are some statistics here that we can go back to. It's telling you that the percent divided is 77.1. So that's saying that from the starting population, 77.1% of those cells have divided. Okay. Drop my pencil. Then you have the proliferation index, and you also have the division index, okay? I'm gonna go back here to our slides so that we can get a better definition. So you have the division index, which is actually the number of cells, cell divisions, or the average cell divisions that the cells will go under And it's taking into account everything, including undivided, okay? The proliferation index is actually going ahead, and it's telling you, the proliferation index, it's telling you the number of cells that have divided, not including the undivided. So, or the average number of replications, rather, I should correct myself, average number of replications for the cells that have divided. So you have to ask yourself, what statistic do you need from this? Okay, do you need to understand from the whole population, including the undivided cells, the average number of divisions, or do you need to understand just from those cells that did start to proliferate, what's the average number of divisions? Okay, the CV typically is somewhere between 4 and 7. Routinely for proliferation data, we're pretty close at about 0.3. Again, that peak ratio, I mentioned it already, it should be about 0.5 or so. That's ideal. Okay, so that's the proliferation modeling. I do have another example from Stella, like I said, that is the cell trace violet. The reason I bring this up, and I wanted to show you that data set as well, is because it's important to know that it's not just CFSE. It's not just, eFluor 670. There's so many different dyes available to you. And just before I get going into that and showing you that second example before we open up for questions, I'm going to show you a couple more things with proliferation, okay?
Yeah, Kathy, let me take advantage of that comment that you just made, because some people here were asking about the comparison of different dyes, in terms of durability, maybe even brightness or their efficiency. So do you have anything to comment on that?
So as far as brightness or anything like that, that's going to depend on a couple of different things, right? It depends on which fluorochrome you're choosing. It depends on your instrument. It depends on the laser power. It depends on the panel combination that you have, if we're talking about resolution, not brightness. So there are a couple of different considerations there. What I would suggest you to do is build the panel that you have to build, keeping in mind there are multiple options for you with proliferation dyes. I do know that CFSE is pretty bright, and you do have to titrate it down. That's my own personal experience from users that I've seen, that the CFSE can be kind of screaming off the charts. And that isn't always a good thing. Keep that in mind, that that's not always what you want. So be careful and be cautious of that. But there are different options, and I would say explore those options to see what fits best for your needs and your panel, because typically when you're doing proliferation, you're building a couple of other different markers into the panel.
Right. And if any of these are more or less sensitive to fixation?
More or less sensitive to fixation. Okay. That I'm not super aware of. I'm not sure if anyone else in the has experience with that. If they want to chime in, I'm more than welcome to that. To those recommendations, but I don't have that myself.
All I know is that yes, you can fix these samples with a fix and perm kit, but I don't know if there's data showing if some are better than others. Or are more sensitive to fixation or not.
Yeah, and the other thing there that's important to mention, you want to titrate these dyes too, because you want to make sure that as you're doing this, you're titrating, you're optimizing for your assay and for your cells to make sure you're not overloading and causing cytotoxicity. And the other thing that you have to be cautious of that, we want to be mindful of is that there might be certain scenarios where the cells pump this dye out. And you might have to take that into account as well. So know the cells that you're working with, know the biology, understand that you might run into circumstances where the cells are pumping out, and you might have to treat with a certain drug to reduce that, to be able to get reliable data. So all of this does come with some caveats, and we'll absolutely help as much as we can with helping you out with that.
Right. So another question here, and I think this is an interesting question. Would the bright bead CV from QC be an accurate value for CV of the proliferation populations? I would say no, it's not an accurate, the CVs that you find in the beads are going to be different from what you find biologically,
Yes.
due to the different distributions of the dye. But it definitely, you want to do QC, just like you mentioned, Kathy, right, with the cell cycle. You want to have the sharpest CVs as possible. So you do want to run, these beads as a QC to make sure that your instrument is performing well.
Yes, absolutely. Yeah, and the reason why you might not see, or won't see rather, the same CVs and beads as you will in cells is because those beads are typically hard dyed. They have a very specific amount of emission that's given off that allows it to be wonderful for us to QC our instruments because it's so nice and tight, and we know that we shouldn't expect variation in those beads. So we have a nice tight signal, a nice tight coefficient of variation. Whereas cells are not beads, right? They're not hard dyed. They don't have an exact number of molecules on there of light that are given off or anything like that, or photons of light that are given off as soon as the laser hits it. So you have to be thinking, all right, beads are beads, cells are cells, they're going to behave differently. It's a great question, but the CVs are absolutely going to be a bit different.
Right. So if we have 100 cells that are all the same, they wouldn't take in exactly the same amount of dye, and that distribution or that variation would be typically larger than you would find in a bead.
Yes.
Hard dye bead.
I want to show a couple of other things, and there's one thing I left out of cell cycle, but it's very easy to show you with proliferation as well. So I just want to show a couple of different things before I show you the next data set, just to show you the cell trace quickly. So we already talked about the model sum and the peak numbers, right?
Sorry, yeah, because I think there's a couple of questions that I think are important before you move on. One of the questions is, why do you count, or why you have 6 peaks that were chosen rather than 5 that were counted?
That's a great question. What winds up happening at the end is, you see how it kind of tails off there? So I use my hand, but you can see kind of there. This would not be one peak, and it starts to get a little bit harder at that low end to resolve. So what you want to do is you want to say, all right, this is the number of peaks that I can very clearly define, and then typically add 1 to that, because the reason for this is, you'll see this is not going to be, this is a low number of cells down here, and it's harder for it to resolve. So we have to kind of guide it and say, all right, we know that it's not going to be very easily visible for us, but we know that this isn't just going to tail off like that. What makes it a little bit easier for us to also do to define positive versus negative of that dye, which would have been wonderful, is if we had a negative cell that didn't have that E4670. But we have to remember that our eyes aren't going to necessarily pull out slight differences in those peaks. And I could also show you, if I look at that, I'm trying to see, maybe here. It's going to be a little bit harder. 1, 2, 3, 4, 5. And you can see how these are nice and tight and have that CV of around 3.3. I'm saying it's a nice tight CV. I can't tell you that you would know that that's 3.33. But this population right here, it's very real. And you wouldn't expect this last population just to be a smear and a spread. We assume that that's going to be a similar CV of 3.3, given the way that the cells distribute their data, distribute the dye, and it pulls in half. So here you can see a little bit easier why we would put in 6 and not 5, right? Because we have these cells right here, and that's a lower number of cells that are proliferated and that are at that generation 5. And it's harder for us to see that via a univariate histogram, but it's a little bit easier to see over the dot plot.
Yeah, and why? So what if you don't have peaks that are so well defined? So that's one of the questions here. How do you feel confident in the modeling when you cannot see, the fit, that it fits so well?
That might require some optimization. So if the modeling does not look correct, what I'd actually encourage you to do is understand that there are different, even different packages out there. So there might be different software programs out there that might fit the data a little bit better. There might be biologically or technically some modifications that you can make to your experiment to optimize it a little bit, feel free to send us some data and we'll do the best that we can to help you. Or even if we're struggling with it, we can also reach out to some subject area experts. What I'd first suggest to do is see if maybe another modeling platform pulls it out a little bit better, or try and optimize the assay a little bit via titration of the dye to pull it out.
Right. So it's very important that, you know, when your data doesn't look good, and this is assuming, you know, that you've done everything correctly, sometimes, you know, biologically, you just don't get that, those nicer CVs like you're seeing here.
Yeah.
But if it's something, you know, that you can improve in your sample prep, and even during acquisition, then definitely it's much more important to focus on that and then get better data, than to try to then model, you know, bad data. Let's put it that way, right? Because the models have their
limitations.
Limitations, exactly. So it's going to be, you know, you can't ask for for a miracle if the data doesn't look that good. But yeah, Kathy, you're absolutely correct. You need to try and see which constraints, etc., which parameters of the model give you a better fit, and look for that. That's your criteria, right? How good my fit can be, and then just go with the best fit.
Yeah. And my first roots of optimization would be take a look at your titration and take a look to see if your cells are actively pumping that dye out, whatever it is. And you might have to look at some reagents that you can add or drugs that you can add to prevent that. I know that I've had experience working with polyploid cell sorting of live cells of hepatocytes for some users, and the name of the drug is escaping me at this point, but you know, we used host to be able to, or the researcher used host, I sorted them, but just worked with them to optimize their assay without the addition of a certain reagent to prevent the efflux of the dye. We were not able to see the ploidy populations at all. And that was, you know, more for cell cycle and not proliferation, but a similar, can't think of the word. it's a similar idea that we want to prevent this dye from escaping. Okay. Let me go ahead now and show you how to export some of these stats. And again, I want to stress that I didn't show you this exactly for cell cycle, but you would do it in the same way. So first, what I'm going to do in here, because I'm kind of done with this proliferation window, once I select create gates. I kind of love doing that because once we have that population and once we create the gates, it automatically gates those populations automatically for us in the workspace. And I'm going to show you why I like that. Okay, I'm going to close out this window. I'm going to open up my table editor. And what I can do is I can drag this table editor down and I can drag in the proliferation node. Once I do that, it goes ahead and it gives me all of the statistics that I might want for the proliferation. If I had this applied to a multitude of samples, what it would do when I created the table, it would give me all this information and give me all of these stats. What statistics you decide to use is up to you based on the biology of your experiment. If I was in the cell cycle experiment, like I previously had been, I just drag in cell cycle and it would give me the same information, okay? So I'm gonna close that out. I just wanted to show you that that's an option. What I can also do is I can come into the layout editor, okay? I can come in here and I can drag in that proliferation. It's also automatically going to go ahead and it's going to give me the report where it's telling me, it's showing me all the populations, it's showing me the modeling, and it's showing me the stats. We've discussed this in previous sessions, but I can come in here, I can change the annotation, and I can call this proliferation dye efluor 670. I can go ahead and I can change what's showing up, the fonts, all of that. I can make any changes here. The other thing that I like showing based on this is I can go ahead and I can drag in proliferation population 0. Okay. I'm going to expand this a bit. And I know everyone loves a good overlay, so that's why I wanted to show you. And then I can show you the population 0, and then if I hit shift and I drag down and I take a hold of all the rest of the populations and I drag them in, it's actually showing me all the different generations from undivided to generation 5 in an overlay. Again, we've discussed this in previous sessions, but if you right click, sorry, again, this is a phone I could not unplug, unfortunately. If we go to histograms and we go to half offset, you can make changes. You can make changes to the coloring and the weight and the style, stagger offset. There's all these different options, but by doing that proliferation, whoever just called got a nice little lesson on proliferation. Maybe it was a telemarketer. You can go ahead and you can make those changes to that overlay. But I really like that you can create those gates just like you did in Cell Cycle. Okay. Now I'm gonna also show you briefly in a new workspace. Save this one. I'm just going to show you what the cell trace violet looks like, and then I can go back to some of those slides and we can review anything that you guys are looking to review after that. So new workspace, I'm going to go ahead in, and this sample that we have for the cell trace violet, it only shows the proliferating data. So, you know, I'm bringing it in to show you an example of another fluorochrome. We would ideally want the unstim control for that. But just to give you an idea of what another dye looks like, okay? And again, thank you to Thermo for providing this. They were very kind in doing that. So I'm gonna double click and open this up. And the other reason this data is nice is because, oh man, all of the events are on the chart edges. Why is that? We're gonna use that handy dandy little option right here for this little circle so we can look at the properties. And what I see is it's from the attune, and the range here is about 16,777,000. Some of our people that are joining us will remember that's also what the range is for the Cytoflex, right? With the APDs on there. So I'm going to come to the transform button. I'm going to customize the axis. I'm going to go to 16,777,000, and I'm just going to set it, not the exact number, but close enough. I'm going to do that for forward and side scatter. Once I do that, that looks much better, right? We're much happier with that. So I'm gonna go ahead and I'm gonna gate here on my presumed live cells. These parameters weren't labeled, but I do know quite a few of them are. So I'm gonna double click on that scatter, and then we're gonna look at forward scatter height versus area. Come in and customize. And for anyone that wants me to go over changing this in the preferences, please let me know and I'll do that after. We did go over that in the intro session. And those intro sessions are available on our website, and they're also posted on our YouTube account. Okay. Single cells. Now I do know that the FL1 channel was FTSE for CD4, or not FL1, that's an old way of talking about it. It's the blue 1 channel in area. So I'm gonna go ahead and just put a population right there, and I'm gonna call those out as my CD4 positive. Okay. And when I double click on that, it's the cell trace violet. So on the attune, that's off of VL1. And you can see here, I'm just gonna go ahead and customize this axis and adjust the width basis a bit. What we're able to see here is we're seeing these populations, but again, we can't look at it just based off of a histogram and set gates, or anything like that, or rather a histogram and set gates. The other thing, I adjusted the width basis, but what I also have to do, if I customize that again, I want you to remember the max is 16 million, and it's not this value of 1 million. So I'm gonna go ahead and change that. And you can see the data is not off scale, which is what we wouldn't want. We wanna make sure that's all on scale, okay? So if I come in here and go to workspace, or sorry, tools, biology, and I'm going to do the proliferation modeling again. Remember what happens if it doesn't go to the right parameter initially. You can go ahead and select this drop down and change it. So VL1 doesn't look great when we start. What I can do is I can count the, oh, What I didn't do, this is actually a great little mistake I made. Well, no, I didn't make a mistake, rather, sorry. I threw myself for a loop. For those CD4 cells, what we see is we have our undivided right here, which I'm going to make that, I'm going to say that that's my undivided, assuming that I had an unstemmed control that I had set right there. And I'm going to count 1, 2, 3, 4, 5, 6, 7, 8, roughly 8, and I'm going to set that number of peaks to 9. Okay, and we're going to look at that root mean squared, and it went from 20 to about 3.63. Okay, now this little peak right there, it's likely at about, that's likely our another peak, so I could always go ahead and add in 10 with that assumption, but I want you to be very careful that this is the reason why you want to go ahead and you want to make sure that you have an unstained. This way you can define your background, okay? The reason I brought this data in was to show you that there are different options, to show you how beautifully it pulls out. But remember, we always want the appropriate controls because we need to define where's our background, where's the positive versus the negative, and also where's our unstimulated. And you can see this data pulls out nice and pretty beautiful without that much in the way of modifications, and the root mean squared is about 2.85. Okay. So just wanted to show you that another example of proliferation data quickly, and how pretty quickly we can get it right in there. I would need to maybe hone it a little bit with the appropriate controls, or I would definitely want the appropriate controls, but then I could hone it and model that a little bit better. And I'm going to close that out. And then I just want to show you briefly, if everyone wants to stick around, you know, an example of the numbers that we get from proliferation. Cell cycle is pretty straightforward, so we don't really need an example on that. But if we go and we look at the proliferation stats example that was provided by FlowJo, if we look at the division index and the proliferation index, what I mentioned earlier is that the division index is the number of times you would expect a cell to replicate if you took into account the cells that had not divided as well. The proliferation index is an example of a stat where you're getting the idea of how many times the cell would divide if you're only taking into account the cells that have undergone a division. Okay, so it's telling us here, I can actually go ahead and do the slideshow now. You have 15,888 at G0, undivided, generation 1, 2, and 3, so on and so forth. How you understand the number of cells at the start of the culture is you do the undivided as is, right? Because that's how many there were to start. And then as you're looking at the divisions, if there's 32,000 roughly in that division 1 population, or generation 1 rather, the starting would be divided by 2. Generation 3, 4, 4 divisions, or divided by 4, and so on and so forth. And I have an example of that on the next slide. Okay, sorry about that. So you have an idea of the total number of cells that you started off with, okay? So the division index, what we're gonna do is we're gonna take the total number of divisions, sorry, again, divided by the total number of cells at the start of culture. So that's why that number is 0.66, because we're dividing the number of divisions by the number of starting cells. For proliferation index, to get a better understanding, we're taking that number of divisions and we're dividing it by the number of cells that actually went into division, okay? So that's going to be the number of starting cells from the culture with that initial calculation of 35,872 minus the number of, 15,888, which brings us to 19,800 or 19,984. And by getting that number, we're then able to calculate the proliferation index, which is higher, and that's 1.18. Okay? If you want to understand how we got the starting cell numbers a little bit, I just made a little slide that people might enjoy, might not, I'm not sure. Hopefully you find it useful. But the starting, right, if we had 1,000 in that parent population of undivided, then if you had 5,000 in this division 1 or generation 1 population, you divide that by 2, so that would be 2,500. Division 3, it's a typo, or generation 2, it's a typo, would be divided by 4, right? So if it's 8,000, it would then add 2,000. And then so on and so forth, right? So as you go along, you're making all those calculations, and then you add all of those end numbers, all those numbers together, and what do you wind up with? You wind up with 15,770 starting cells. I just thought drawing that in there and getting an idea with the calculations and why we're doing what we're doing might make it a little bit easier for people to understand. And feel free to use those references. Take a look at the slides that I send out, and also take a look at FlowJo's website for a detailed description of some of those statistics. And also know that in the slides that I send out, we will have a reference to a paper by Mario Roeder that discusses some of those statistics. So please feel free to reference those to be able to get a deeper understanding as well. And I think with that, we can open up for any questions. You guys have stuck with us again. We went past an hour again. I think it's pretty safe to say we're probably going to be about an hour and a half each time or so. So yeah, open it up to any questions, and thank you guys also.
Thank you, Kathy. Wonderful again.
Thank you.
So there's a question here that regarding the unstained control. So if you have the unstained control, would you gate out the autofluorescence first and then define the peaks? How would you do it?
So if we have the unstained control, or what I would ideally like for proliferation is to do a gating of the scatter single cell CD4 and then have a sample that's truly an FMO, right? So have an FMO sample that doesn't have that proliferation marker in it, whether it's CFSC, whether it's an anti-BrdU, whether it's a cell trace, whatever it is. Take a look at that population. So say, for example, I don't know if this is going to work. No, it's probably not going to work. Say, for example, I can't do it at this data set. I would take a look at the population that's gated on the cells of interest, and then from that population, I would go ahead and I'd set a gate. So say this is where my negative cells are. And say this is a negative based on an FMO, okay? What I can then do, if this was the channel that I was using for my proliferation, I can come into workspace, statistics, and I can go to BL3, 6,187. It's a very high number because that's not really what we're looking at here, and I'm just showing a mock-up. And then I can fix that background at 6,187, and the modeling software would understand that anything, it needs to be above that value to be able to be considered positive for that proliferation die, if that makes sense. It's not a great example for me to be working from, but just to give you an idea.
Right. Right. So there's another, so one of the questions was, so regarding, to going back to the cell cycle data, do you suggest using the old style DNA QC particles like the chicken erythrocyte nuclei or the cow thymocyte nuclei as biological controls for the cell cycle experiments?
Oh, sorry.
yeah. But you know, because so the comment here is that they can be used as biological controls for staining, for double discrimination, CVs, and linearity. So to set up the instrument essentially.
I've tried working with those before. They could be a little bit tricky to work with. I don't know if it's just in my hands because it's been a little while since I've been on the bench. Staining samples and everything like that, I do it intermittently rather. I don't necessarily suggest those to our users. I've tried them. Typically what I ask is for a nice cell line control. That's something that we have users that come in to do sorting. They have their tissue that they are using to see a variety of different populations, but they have their cell cycle and a cell line is a nice control for us to be able to see you know, that we're in a linear range, that everything looks nice and beautiful, and our peaks are nice and tight. So that's usually easy enough to have a nice cell line that you can, like a nice healthy cell line that'll have a normal distribution of G1 and G2. And I think that should be sufficient. If you're having a hard time with that, you can always explore, you know, the chicken red blood cells and everything like that, but it's not something that I typically tend to use.
Mm-hmm. Okay. So actually a question here from one of our FlowText partners, Joel Sederstrom. He was asking if whenever we look at the attune data, do we look at height instead of area? Because it seems to, you know, to look better in that way. I was actually looking for our sales rep, our thermal sales rep, but I think she left just as he asked that question.
Okay. Joel, that's an awesome question. I have to be honest that I've never worked on an attune, and I just got this, this FCS file from Thermo. We can take a look at the height and see if it pulls it out a little bit better. So if we open that up and take a look, you can always explore that. I just haven't had much experience on that instrument. So if we open up the negative and we take a look at the VL1 in height, let me just adjust that axis there as well. 16,777,000. That's the same. I have to adjust the, sorry, width basis as well. We can see how the CUVs look there. So it was about 3 point, on our VL1 in area, it modeled it with a CV of around 2.85. And our VL1 in height, it modeled it the CV of around 3.52. I could take a look at the stats and go in and draw some gates and see which one pulls out better, to try and understand it myself, but I don't have too much experience with the attune. It looks like both are modeling the way they should, but there might very well be a technical aspect to preferring height for the attune.
In some cases, yeah.
Yeah.
Right. Another person was asking about the cell numbers for FMO controls. Do they have to be the same as that of the samples?
It all depends, right? So if you're trying to get an N population, if you're just looking at the CD4 positives, you really shouldn't have that much of an issue getting a nice robust population of CD4s. If it's a nice healthy sample, to be able to get maybe 5 or 10,000 of that gated population. When possible, it's nice to have the numbers match up to be able to get an understanding of that spreading. But it's not going to be, for this application, super, super critical, right? If you have 10 cells, it's not going to be ideal to set and get MFI values off of a very small number. I would at least recommend a couple of hundred, preferably a couple of thousand cells gated for the FMO of that final population to be able to define the background. But does it need to be the exact same number? Not necessarily for this application, though.
Right, not necessarily, but you know, one of the things that's really important is, so when you're labeling with antibodies, you have to make sure that the number of antigens is relatively the same. But
yeah,
it's much more important in a DNA cell cycle experiment or even proliferation.
Critical, yeah.
Right, to have the same number of cells when you label them, right? When
yeah, absolutely.
Like, in this case, the FMO doesn't, you know, if it's the FMO without the dye, then it's not as critical. But as in best practices, yeah, it should be.
Yeah,
it should be right, staining all the same number of cells.
Yeah.
But it's not, not critical as long as the number of cells doesn't change too much.
Yeah.
The other question is if BrdU PI cell cycle analysis is always better and more accurate than PI only cell cycle analysis.
So what I can do for that, that's a great question. I actually wanted to show that last data set. So for anyone that's sticking around, I think we still have a couple of people. What I can do is I can show you that data set, and I'm going to go through it very quickly to not waste people's time. I'm going to bring in this bivariate data. Very quickly going to show you why I think that I would suggest whenever possible to look at, well, I keep getting that coming up, sorry. Whenever possible, I would suggest going ahead and taking a look at the BRDU in combination. Pull them. Scatter. Drag that up. Okay. This one does not have, this is an old FCS2 data set that was kindly provided to me by John Quinn from FlowJo. So he was great in giving it to me. I just can't gate for singlets, unfortunately, which would be important here. But if I just look at DAPI and I look at a histogram and I look at a nice control here, I'm going to go ahead and I'm going to look at cell cycle on that scatter population. So it was grayed out because I didn't select the population. So I'm going to select cell cycle. All right, very quickly, let's look at our RMS, our root mean squared. It's okay, could probably be a little bit better. For our sake, this is not too bad. I'll try and optimize it a little bit if I can. Constrain the G1. I'll set the CV. Maybe try and optimize it a little bit. This needs to be about 10. Nope. But again, this RMS actually is not too bad. The G2 peak, and I can come in, I can play with this a little bit. 1.7 is way too low. 1.9 looks a little bit better. Seeing if I can just make that a bit better, but I think it's actually okay where it was. I'm just trying to make the modeling as best as I can in a brief period of time to show you why I think BRDU is better. So let me go ahead and bring that cell cycle up. I'm going to go along the RMS. It's okay. Let me use this one for an example. So this RMS is showing us it's acceptable. Our G1 percentage is 40.5, our S phase is 34, and our G2 is 18.1. So I'm going to drag that along to the side. Modeling could be a tiny bit better, but for the most part, some people might see this, or some researchers might utilize this modeling. But if I take a look at this scatter and I look at DAPI on the x-axis and I look at BRDU on the y and I change that to log, this is our control without BRDU. It was just a little bit of a typo there. And then when I go to the next sample, which is this one right here, it's 1 hour, tube 1. I can very clearly define my populations. I'm not going to include the polyploid or anything like that. I'm going to be a little bit stricter here. And I did want to show this, so thank you for the person who asked us that question. I should have put S phase there, but everyone knows what I'm gating. And I could use a polygon too, but I'm just doing it for sake of time. And because I tend to be a little anal, let me change that to S phase. Oh, well, you all get it, S phage. Okay. When we look at these percentages, what do we see? G1 went from 40.5 to 27, okay? So even though it's giving us a pretty nice distribution, the actual percentage of that G1 is a little bit off there, right? In this modeling. And what we're seeing by the incorporation of this nucleoside of BRDU is we're seeing that that S phase is actually closer to about 50% as opposed to 34. So this is a really beautiful example of why we suggest for our users to incorporate you know, another parameter into their cell cycle experiments to be able to more clearly identify through 2 parameters their different stages of cell cycle. So sometimes you pull it out really beautifully, other times it doesn't pull out as nice as you'd expect with Univariate. If you're questioning it, please go ahead and add in the BRDU or EDU. Okay. Is that a good example, Rui, you think, or?
Yes, I do think, yeah, it's a good example. I mean, it does show that BRDU, if you can pull it, the S phase apart, you do see from that BRDU plot that there is a huge overlap between, you know, some cells that would be considered G1 or G0,
Yeah.
And some cells that will be considered G2, but they're actually in DS phase.
Yeah.
And then if you look at the fitting, you do see that overlap. You know, when it models, it shows you an overlap between those populations. So BRDU really pulls that S phase apart and enables you to get a much better discrimination of these phases.
Yeah, absolutely. And, you know, I could have spent a little bit more time trying to optimize that modeling a bit and reduce that root mean squared. But I think regardless if I had spent 10 minutes on it, 5 minutes on it, whatever I spent, I still think, or I still know, I'm not even going to say think, because I'm incredibly confident in that the addition of that BRDU is going to help us, you know, more than we can imagine.
Yeah, so I have a question here that, you know, the person is saying, maybe this is a stupid and not technical question regarding the proliferation analysis. Yeah, exactly. There's no stupid question. Regarding the proliferation analysis, why divided cells display multiple peaks? Is it because that the dye is only in the parent cells and the concentration of the dye gets diluted every time the cells split?
All right. Maybe this might seem a little bit silly. I'm going to try drawing something because I like drawing stuff out to show people. I'm going to hold it up to the camera as I talk about it. So say you have one parent dye and it has, I'm going to draw a bunch of X's, and those X's are going to equate to fluorochromes. So this is, I'm going to draw a parent, okay? I don't know if that comes up backwards, so I apologize. But that parent has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Nice, I drew an even number. 10 molecules of that dye. Then what winds up happening is it splits. That parent then becomes 2 daughter cells. Now, what winds up happening? There's a limitation to the number of to the dye that's in there. So when that goes, and that goes to 2 cells, what do you have? You have the daughter 1 that has 5 and the daughter 2 that has 5. What would that equate to when we're looking at MFI? This is one event that has double the amount that's in these 2 events. So if I had a histogram then, and we looked at count on the Y, because it's a histogram, and we looked at intensity on the x-axis, and say the count was just 1 or 2, you would have an intensity. So this population, this parent population, would be the higher intensity. And this, if it's backwards, I apologize. And these 2 right here, which have half the amount, are they're 2 count, right? And that's going to show up a little bit lower in intensity because each of those individual cells has half the amount of fluorescence as that original. So it's doubled in number, but it's half the number of fluorochrome that's in there because of the division of the cell. I hope that if that doesn't clarify it for you, I'll try and
Yeah, no, I think it's clear.
Okay.
Very clear. Yeah, but I think that's it for in terms of questions. Yeah.
Okay, awesome. We'll go ahead and we'll, again, we've recorded this session, so we'll upload it on YouTube today. We'll get it out to everyone. We'll share the slides with everyone who registered, regardless of whether or not they attended. And we're working on next week's topic. So hopefully this week will be a little bit better about getting out to you what our topic's going to be for next week. Remember that Zoom link that we're sending out, if you already registered for future weeks, you're good to go. If not, that one Zoom link is good for registration for whatever day you're looking to sign up for. But we're going to continue to send that out in case anyone's forgotten to register for any days, and we'll distribute all of the information. So thank you. I hope everyone stays safe, and thank you for being patient with all the phone calls and everything like that, and have a good day.
Okay, thank you, Kathy.
No problem. Thank you.